The movement of solutes across a selectively permeable membrane. Experiment 2, Part E. An introduction to this experiment. Diffusion is a spontaneous reaction. Diffusion does not require energy, and diffusion is always occurring. Facilitated diffusion is also a spontaneous reaction, which does not require energy. However, it requires the help of transport proteins such as channel proteins. Now in this picture, you can see a high concentration, low concentration. Diffusion is occurring in both ways. However, the rate of diffusion from high to low is higher than the rate of diffusion from low to high. So the net rate of diffusion is seen as high to low, and over time, this rate of diffusion will result in an equilibrium as seen here. Now this also displays the second law of thermodynamics, which states that everything is moving towards entropy or disorder. Cells move towards entropy or equilibrium. Equilibrium representing the highest state of entropy or the highest state of disorder. A cell at equilibrium is a dead cell. The cell uses many concentration gradients, as seen in the picture here, which result in highly organized spaces and also potential energy. Here we see a channel protein, a high ion concentration with high potential energy in these ions, and a low ion concentration. These ions are more likely to move from here to here and will move from high to low until equilibrium is reached. Factors that decide whether a molecule can diffuse through a membrane include polarity, size, and charge. In this lab, or in this experiment, we are looking at the factor of charge, as small molecules or ions can diffuse without any help, and large molecules need proteins to help, as in facilitated diffusion. And in this lab, the dialysis tubing is not going to have these proteins to help, and facilitated diffusion will not occur. Only the small molecules and ions which can diffuse through the pores, those are the only ions or molecules that will diffuse. Polarity is due to the bilayer structure of membranes, the inner nonpolar lipid tails, and the outer polar heads. Charge can also affect whether a molecule can diffuse through a membrane. The procedure of this lab is the filling of a dialysis tubing with a starch Na2SO4 solution, and this tubing, the dialysis tubing, serves as a plasma membrane. Now, obviously, the tubing is not as complex as the phospholipid bilayer found in cells. However, it still acts as a selectively permeable membrane and will get the job done. The sodium sulfate is the intracellular materials. We submerge this tubing into the beaker solution, which serves as the extracellular environment. The beaker solution was albumin in a aqueous NaCl mixture. Albumin is a protein, NaCl is an ionic compound, and I'm just going to go back real quick. Na2SO4, that's an ionic bond between the sodium and the sulfate, and a covalent bond between the sulfur and the oxygens. We let this rest for 45 minutes. We submerged the tubing in the beaker solution and let it sit there for 45 minutes. Then we dispensed the tube solution and the beaker solution into these respective test tubes. Test tubes 1 through 4 contained 5 ml of tube solution, and test tubes 5 through 8 contained 5 ml of the beaker solution. We performed the starch test on tubes 1 and 5. 1 is the intracellular, or the cell, and 5 is the extracellular. The sulfate ion test, again, on the square here, is the cell and the extracellular. 2 and 6, 3 and 7, 4 and 8, respectively. Each test was conducted once on the inside of the cell and once on the outside of the cell. And we noticed the color changes of the solutions in the tubes after adding in, for example, Bure's reagent for proteins. And we had done this before in lab 3, where we tested for glucose, starch, lipids, and proteins. This is the setup of our experiment, or the second part of the experiment. As you can see, tubes 1 through 4 are the cell, and tubes 5 through 8 are the extracellular fluid, after letting everything rest for 45 minutes. Before conducting this next part, we hypothesized that starch would not diffuse across the selectively permeable membrane. In this case, the selectively permeable membrane was only focused on size because of starch's large size. So again, starch would not diffuse because of its size. We hypothesized that sulfate ions should diffuse because they are relatively small. We can calculate the molecular weight of sulfate ions and arrive at the number 96 AMU, which is relatively small. Hence, we decided it could fit through the pores and it would diffuse. Chloride ions, even smaller than sulfate ions, with an atomic weight of 35 AMU, and obviously these are going to diffuse. Proteins, however, would not diffuse because of their large size. In this case, albumin, it's a huge protein. It's not going to fit through the tiny pores in the dialysis tubing. The starch test. Starch was present in tube 1 as seen by this dark blue coloring here. Starch is absent in tube 5. It's a negative test. Starch originated in the solution we put in the dialysis tube, which we then transferred to tube 1. Tube 1 is the cell, and tube 5 is the extracellular fluid. Starch started in the cell and never diffused to the extracellular fluid because it is a large carbohydrate which could not fit through the pores in the tubing. Sorry, let me go back. If we let part 1 run for a longer amount of time, from 45 minutes to maybe a day, there would be no difference in the results. Starch is always going to be too large to fit through those pores. The sulfate ion test. Sulfate ions are present in both tube 2 and 6, both in the cell and the extracellular region. Matches our hypothesis. Both tubes show a white precipitate. It's hard to see in this picture, but they both these tubes were tested positive. However, you can tell that the amount of precipitate is different in each tube. This suggests that equilibrium was not reached. Tube 2 contained the cell, and the original cellular fluid was the origin of the sulfate ions. Now, these sulfate ions were small enough to diffuse, but the rate was not great enough to reach equilibrium in 45 minutes. Hence, it is the amount of precipitation is different and is greater in tube 2 than in tube 6. So, if we ran part 1, resting for 45 minutes to maybe a day, equilibrium would have been reached. The chloride ion test. Chloride ions are present in tube 3 and tube 7. Both tubes show the silvery precipitate seen in the circles here. They both were positive. The cell here and the intracellular space the extracellular space, sorry, in tube 7, because the amount of precipitate and the consistency is similar in each tube, we concluded that equilibrium was reached, and therefore if we ran part 1 for longer, there'd be no significant change. Now, we could assume, or some might assume, that there's no diffusion occurring after the 45 minutes. However, there is diffusion occurring. The equilibrium is just maintained, because equilibrium, again, the second law of thermodynamics, everything is moving towards disorder. Disorder is favored, so we are going to maintain an equilibrium. Tube 3 and tube 7, again, similar parts, similar amounts of precipitate, hence they are have reached an equilibrium. Tube 7, now, the extracellular space contain the chloride ions in the form of NaCl, and we're going to talk about why NaCl dissociates and why Cl could travel while Na didn't in future slides, but the chloride ions are small enough to diffuse and the rate was fast enough to reach equilibrium in 45 minutes. Protein test. Protein is present in tube 8, which is where it started, the extracellular, the NaCl albumin being the protein, that's where it started. And there's absent in tube 4. We can conclude that equilibrium was not reached because albumin did not even diffuse across a selectively permeable membrane because of its size. Now, if we ran part 1 for longer, there would be no significant change. Again, the size of the albumin is not going to change, and it's not going to be able to diffuse through the tiny pores. So without a facilitator or a channel protein, which is not present in the dialysis tubing, we're not going to see any diffusion of protein, even if we ran part 1 for longer.